Welcome to our new abberior webinar. My name is Jasmin and I'm a social media manager here at abberior. And I think it is great that you are joining us today to learn more about novel techniques and dyes that improve live cell super resolution imaging. We have two great talks coming up today which have been pre-recorded. We will have a live Q&A session at the end of this webinar. So if any questions come up during the talk, please drop those into the chat box and we will come to this later in the discussion. Perhaps you have asked yourself, if the resolution of a microscope is constantly evolving, how do the fluorophores and probes that are used for this purpose keep up? These and many other questions will be answered by our two experts from abberior. So I'm happy to introduce to you Kristina Kramer, who is an application specialist here at abberior, and she was involved in the testing and optimizing of our new probes. Hi, Kristina.
Hi, Jasmin.
Also here with us is Florian Grimm. You might already know him from the past webinars. He is an old hand in business, the first employee in the company and an absolute professional in the development of dyes. Hi, Florian.
Hi, Jasmin.
So recently, abberior has launched two new products, which improves live cell super resolution tremendously. As you know, the red to infrared dyes come with many advantages, like improved resolution and low phototoxicity. So what is so unique about the new abberior Live Red?
The most commonly used dyes for live cell application are rhodamines or rhodamine-based fluorophores, and they can exist in two different forms, in an open form and a closed form. The open form has a switterionic character and is fluorescent, but because of these two charges, the dye cannot really easily enter the cell. But with the closed form, although it's non-fluorescent, the dye can pass the cell membrane just by diffusion. And it's very important to tune or to play around with this equilibrium between the open and the closed form. And there are many techniques available. And one technique has been developed a few years ago by Gracidas and his group here in Göttingen at the MPI. And they introduced a new isomeric form and also a neighboring group effect. And if you are more interested about it, you can scan the QR code. We already had a webinar with him where he explained this very specific isomeric form and the technique. But very recently, his group also showed a new synthesis method to tune this or to push this dyes into the red region, into the red and infrared absorption and emission region. And we from abberior are now able to use one of these recently published dyes as our new abberior Live Red dye. And Kristina can now show you some examples and impressions.
So we are able to offer now the new Live Red dye as labels for tubulin, actin, SNAP and DNA for live cell imaging. And this offers very nice results in STEP microscopy. So as Florian already mentioned, due to the open and closed form equilibrium of the dye, we have a high cell permeability of the labels and also a very good biocompatibility, which means that we have no or low cytotoxic effects and nearly no effect on the living cell and its metabolism. Moreover, the new Live Red is a very good partner for our labels carrying the Live 590 dye and also the Live 460L dye, which work together in combination very good with the red stat laser at 775 nanometer. So in the lower left corner, you see a very nice three-color staining of a mammalian cell that expresses a SNAP-tag fusion protein that decorates the outer mitochondrial membrane. And this is labeled with our novel abberior Live Red label for SNAP. And in combination with that, we have labels for actin and tubulin, which is the Live 590 tubulin label and the Live 460L actin label. And these work in combination with one stat laser, which is at 775 nanometers.
Thank you, Florian, and thank you, Kristina, for the nice introduction of the new abberior Live Red. So next to the fluorophore, the label is also important to improve your live cell super resolution experiment. Recently, abberior has launched the new Live HaloX. So what is the difference between the standard HaloTag ligand and the new abberior live HaloX?
So in the conventional HaloTag technology, it is based on the expression of the fusion protein of the protein of interest and the HaloTag protein. And in the common approach, there is the formation of a covalent bond between the fluorescent label and the HaloTag protein. In the HaloX technology, which was developed from Kai Johnson and his group, we have not the formation of a covalent bond of the label, but a non-covalent interaction of the fluorescent label and the HaloTag protein. And this allows a constant exchange of the fluorescent label at the protein binding site. So thereby, photobleaching is reduced, as bleached labels can be exchanged at all the time with unbleached fresh labels from the reservoir of the surrounding medium. So this enables also multi-frame stat imaging with very low photobleaching and also extends the platform of HaloTag to methods like MinFlux and Paint.
Okay, so we tested these new probes also here at abberior and came up with this new 3 products, which we call abberior live SPY 555, abberior live 590, and abberior live SIR HaloX. And we also tested these probes on mammalian cells. So we received cells from Kai and his group expressing TOM20 fusion protein with a normal HaloTag 7 protein, and tested these probes on the cells together with our direct labels. So on the upper left corner, you see a 3-color stat image, for example. And we also tested the probes in time-lapse experiments, so where we stained the cells with these probes and imaged multiple frames over the same region of interest. So in this example, we imaged 60 times in a stat mode, and what you see here is a raw data. So you see almost no bleaching effect, which means that this exchange of the fluorophores has a very high impact. And those probes are also able for fixed cells, not only for living cells, so you can use it, for example, for Paint and MinFlux. And I think this already shows you that this technique is very cool and outstanding, and I'm pretty sure that Julian will then later on present us more details about this new technology and this new probes.
Thank you, Florian. Thank you, Kristina, for the nice overview, and I believe that these 2 products will push live cell super resolution imaging to the next level. So here you see a QR code. Please feel free to scan it. It will lead you directly to our labeling protocols that we have used for these images that were just shown. So for more details, as Florian mentioned, into the HaloX technology, we have Julian Kompa here with us, who will present his data. I am now standing here with Julian Kompa. Julian is a PhD student at the Kai Johnson Lab at the Max Planck Institute for Medical Research in Heidelberg. He has been significantly involved in the development of the new HaloX technology. Julian, I'm happy to have you here.
Hi, thanks for having me.
Thank you. So, as mentioned, you're working on your PhD. What are the main questions you would like to answer with this project?
So, fluorescence microscopy is technically non-invasive. And this brings us into the position that we can observe biological samples in an unperturbed manner. However, everybody who's handled a microscope knows there are still many challenges left. For example, photobleaching just reduces the amount of information that we can gather over time and thereby often precludes dynamic measurements. Then we also need to put our sample under harsh conditions like strong laser irradiations or specialized buffer conditions, and this may alter the structure or function of the cells. And if we then want to observe processes in multicellular organisms, we are often even lacking the techniques to place a fluorophore on a biomolecule. And all of those tasks I would like to address by designing and improving new fluorophores, for example, to have them more photostable or to be less phototoxic. And also I'm thinking about new labeling strategies to place a fluorophore on a position of interest And in the long run, I envision that we have very small modifications on our targets and ideally can do so in living systems in vivo.
Thank you for the summary. So fluorescent microscopy is one of the methods you're using during your PhD. What is so fascinating about super resolution microscopy?
So first of all, I really like to work in this very multidisciplinary field. So improving nanoscopy starts from the engineers and the biophysicists who are building the setups. Then we have chemists and cell biologists trying to tune the labeling. And in the end, we have the whole field of data processing. And it's really a pleasure to have a view in all of those fields and to learn from all of them and being part of this joint venture. And then in addition, it's really cool to me to work with living samples. So just looking at live cells that are glowing and also working on a stat setup where you can visually see the difference between a regular image and a super resolution image and really can observe what is going on in a live cell.
Cool. Sounds really interesting. And we are now looking forward to your talk. The stage is yours.
Thank you. For today's webinar, I would like to guide you through the development of our exchangeable HaloTag ligands and give you some examples how you can use them for super resolution fluorescence microscopy. Modern fluorescence imaging possesses high requirements on the fluorophores and also on our capability to label biological structures with a dye. The dyes should be ideally bright, photostable, and diverse in color, which are some features that are united by a class of dye called rhodamines that together with carbopyranines and silicon rhodamines fan the visible spectrum. Rhodamine dyes are best combined with self-labeling proteins. Those genetically encoded fusion proteins are non-fluorescent, however they can provide a specific and fast labeling reaction with a bio-orthogonal substrate. The most prominent examples are HaloTag, SnapTag, and ClipTag. And for example, HaloTag makes a covalent bond with the common chloroalkane substrate that you can see here. And this then can be attached to a rhodamine dye. However, rhodamines do not only exist in this open zwitterionic and fluorescent form, they can undergo undergo an intramolecular spirocyclization reaction, forming this closed spirolactone, which is non-fluorescent. And this might sound incremental at first, so having a fluorophore that is not always fluorescent. However, this non-charged spirolactone actually improves a lot the cell permeability and is able to enter live cells. Once bound to a protein surface, for example, the surface of HaloTag, the open form can get stabilized, leading to a fluorescence increase upon labeling, which we call fluorogenicity. And this largely helps us to suppress background signal from unbound or non-specifically bound dyes. Taken together, it's not a big surprise that HaloTag and rhodamines have become a mainstream tool for super resolution imaging. It's very established, for example, for live cell stat, but can be also used for single molecule localization microscopy in combination with self-blinking rhodamines. And also the recent MinFlux publication had featured some HaloTag and some rhodamines. However, and that's something you have to keep in mind, the labeling of self-labeling proteins, as for many other labeling strategies, is permanent. And this causes some consequences. First, once bleached, the dye cannot be detected anymore. As you see here in this experiment, using covalent labeling and stat microscopy has some limited amount of frames that can be recorded. In contrast, using dynamic or exchangeable probes that can be replenished by fresh fluorophores once they are damaged allows to acquire much more frames and thereby has the potential to observe dynamic processes. Secondly, multiplexing is limited by the amount of dyes that we can discriminate. every permanent label will block a channel. However, if you think about exchangeable probes, you can image a target, wash it out, bring in a new fluorophore for a new targeted structure, and thereby multiplexing is potentially unlimited. And finally, the transient binding of exchangeable probes is a key concept of paint and minflux. The on-off binding creates sparsicity, which allows us to distinguish between close-by molecules. On the downside, only very few live cell compatible labeling strategies are also compatible with probe exchange. And this in the beginning motivated us to review again the concept of HaloTag, which is covalently labeling the chloroalkane substrate. And by looking at the crystal structure and doing some docking experiments, we actually came up with the idea to replace the terminal chloroalkane chain with an alkyl sulfonamide. This sulfonamide can be either a methyl sulfonamide or a trifluoromethyl sulfonamide, and we called those ligands S5 and T5, and they have binding specificity for HaloTag, but with the big advantage to be exchangeable. Looking again at the crystal structure, we now see that those sulfonamides nicely occupy the binding site and make some polar interactions with the key residues. We next ensured that the labeling is non-covalent by performing in vitro protein labeling experiments in contrast to the classical halo substrate. Looking at some key properties, we have a binding affinity of around 100 nanomolar, which is well suited to have specificity for HaloTag binding, but also gives the option to be exchanged with exchange kinetics in the range of paint labels. The biggest difference between S5 and T5 lies in their fluorogenicity. So as you can see here by fluorescence emission spectra in presence and absence of HaloTag, SIR-T5 yielded a similar fluorescence turn-on as covalent SIR labeling, whereas the S5 ligand shows decreased fluorogenicity with a higher background fluorescence in absence of HaloTag. Next, we combined S5 and T5 with a range of fluorogenic rhodamine dyes, mostly from the Max Planck dye series and the Janelia fluor series. And as you can see here, in those confocal live cell imaging experiments, all of the probes delivered specific and bright nuclear signal from U2S cells expressing histone 2b HaloTag fusions. In this experiment, we show the reversibility of the probes in live cells. So the nucleus is covalently labeled with a green probe, and we flush in and wash out a red XHTL. You see that the staining is fully reversible, and in addition, the staining and unstaining takes about 1 minute. In the following, I will pass some super resolution microscopy experiments. And first, we teamed up with Mike Heilemann and Marius Glogger, who performed this paint imaging. This is a fixed U2S cell expressing vimentin HaloTag, and in low probe concentrations, the unbinding and binding of JF635S5 can be seen as blinking. Summarizing a couple of 10,000s of those frames yielded us this super resolved image of intermediate filaments. And we achieved a localization precision of around 11 nanometers, which was comparable to DNA paint control experiments. We did the same experiment basically by using minflux, and here we achieved 4 nanometer localization precision, really resolving single vimentin molecules. Next, I'm coming to some live cell stat experiments. This time we expressed HaloTag with a mitochondrial localization signal TOM20, and we tried to generate as much stat images in a small 10 by 10 micrometer ROE as possible comparing exchangeable and covalent staining. And as you can see here, the covalent labeling bleaches fastly within the first 10 frames, whereas the exchangeable HaloTag ligand provides a much more stable signal, reaching up to 30 to 50 frames at least half maximum signal intensity. This can be also used to perform 3D stat. In this case, we imaged a U2S cell that is fixed, expressing HaloTag at the endoplasmic reticulum, and it allows us to reconstruct the ER at high precision. And this works also in primary cells, in this case red hippocampal neurons, resolving their plasma membrane in 3D. So far we only spoke about one color imaging, but we actually also developed a second system providing dual color imaging. Therefore, we took a variant of HaloTag, the so-called dead HaloTag, which has a single point mutation D106A. This point mutation usually shut down the covalent labeling reaction, but it also has much lower binding affinity for S5 and T5 ligands. In addition, we found out that ligands terminating with a hydroxy moiety, we call them Hi4 and Hi5, possess some nanomolar high affinity binding to dead HaloTag, while not binding or binding much weaker to the regular HaloTag. And the binding specificity difference is around 100-fold. This is demonstrated here again in some live cell experiments where we have again cells expressing HaloTag or dead HaloTag in their nucleus, and you can see nicely that S5 and T5 only bind to the HaloTag expressing cells and much less to dead HaloTag, whereas it's the opposite for Hi5 and Hi4, which do not bind to HaloTag but deliver specific signal of dead HaloTag. And this can be also used in a single cell, expressing both HaloTags at different localizations and co-staining with the two probes that then can be nicely discriminated. We use this system also to perform some dual color paint imaging, in this case of mitochondria and lysosomes, and this is an exchange paint approach. So first we imaged one of the targets, then exchanged the fluorophore and imaged the second target. And this is also compatible with live cell stat imaging, using both probes simultaneously and depleting them with the same depletion laser. And this allowed us also to track the dynamic interactions of mitochondria and lysosomes over a couple of high-resolution frames. At the end, I would also like to take a look in the future, because I think that we can largely benefit by combining different exchangeable labels. And here our collaborator actually did exactly that by synergizing our XHDLs with various paint probes, recording this 6-color stat image in fixed cells. With this, I'm at the end and would like to summarize. We added a new dimension to HaloTag, which are exchangeable labels. They are fluorogenic and tunable in color, and actually they are very easy to handle by just adding them to the cells, waiting a couple of minutes, and then the staining is ready. I've showed you versatile applications for super resolution microscopy in time-resolved and 3D stat in live cells, as well as paint and MinFlux. And it's possible to image two HaloTag variants at the same time by using this dual color system, comprising of XHDLs and HaloTag pairs with different binding specificities. And in the future, I hope that we can synergize exchangeable ligands to overall improve super resolution microscopy. I would like to thank abberior for having me. This webinar is a great opportunity to share our science, and also like to thank all people involved from the Max Planck Institute for Medical Research, Department of Chemical Biology, Kai Jonsson, of course, but also Julian Kompa, Jorik Bruns, Jonas Wilhelm, and Michel Frey, Mirek Tarnawski, and Elisa De Este from our facility, and finally Mike Heilemann and Marius Glogger from Frankfurt University. Thanks for your attention.
Now we have reached the end of our webinar, and we will move on to the live Q&A session. In this session, we will answer some of the questions you have asked in the chat. If not live, we will get back to you by email. So thank you for joining us up till now, and we will see you in the live Q&A session.
Okay, so hello everybody. Welcome now to our Q&A live session. Again, sorry for the delay and the technical problems that we had in the beginning. We hope that you were able to enjoy the talks anyway. And thank you all for staying with us until now. So there are plenty of interesting questions coming in. So let's start with the first one, which goes to Julian. And someone asked, how did you come up with the chemical design of the exchangeable HaloTag ligands?
Thank you. Good question. So I think what helped a lot is that we had already quite some structural knowledge about the probes and also about the HaloTag. And yeah, in the beginning, I did this very naive docking screen where I just tried to somehow fill the binding pocket with something that is not a leaving group. Other than that, we kept the ligand, as it was, or as HaloTag was evolved on it. And something that is also very important to know is that HaloTag has some kind of an intrinsic binding specificity for rhodamines, especially carborhodamines, silicon rhodamines, and that helped already to gain nanomolar affinity in the end.
Okay, nice. So you talked a lot about the advantages and the nice things about the exchangeable HaloTag, but are there also limitations or technical pitfalls that you can talk about a bit compared to the covalent version?
Yeah, sure. I mean, I wanted to highlight that exchangeable probes can be beneficial for some applications, but of course, having a covalent labeling is still very powerful, for example, for pulse chase experiments, for post-hoc analysis. And one thing you have to keep in mind is that we added this bleaching resistancy, but we kind of bring the burden to do no wash imaging. So the fluorophore has to be always present. You cannot wash. This is possible due to the fluorogenicity of the probes, but there will also be some background. There's always some background. And if you try to image some very low abundant proteins endogenously tagged, of course, you have to find somehow a compromise between signal intensity and the background. And then again, you might wish that you can wash. So that's just something that has to be tried out, that has to be seen. Of course, also a covalent reaction over time, but always go to the maximum labeling, whereas exchangeable probes will stay in an equilibrium.
Thank you. Then one question to Florian. Also concerning the HaloTags, there's a question in the chat if these reversible HaloTag ligands are commercially available.
Yes, they are. That is also the reason why we are doing this webinar here, to announce that we have these exchangeable HaloTag ligands now available in our web shop. So there are 3 probes available. One with a silicon rhodamine in cooperation with Spirochrome, then with SPI 555, and then an orange fluorescent dye, so the Live 590 with a HaloX. So there are 3 probes available with this exchangeable Halo technology. Yeah.
Thank you. Another question to Florian. So what is the recommended working concentration of the live red labels that you presented in the beginning? And is there any information about the staining protocol or procedure?
So to answer the second question right away, so we have protocols on our website which you can download. There are also protocols for our antibodies, for other direct labels, for NHS, Easter, and so on. So you can go to our website, to our protocols, and then you will find also for HaloX and SnapTag the protocols. And then the second question regarding the concentration of the live red dye. So you need quite low concentration in a range of 10 to 100 nanomolar, which is quite low. And then stain it for half an hour roughly. And it's really specific and bright. So that is, the reason is because of this high biocompatibility. We can use very low concentration to stay specifically and bright the targets inside the cell.
Mm-hmm. Thank you. Another question to Julian. So you also talked a bit about HaloTag proteins for paint microscopy, and there was one question concerning the fluorogenicity and is whether this is compatible with methods like paint, and maybe also in this context, what makes HaloTag-based paint attractive compared to DNA paint?
Mm-hmm. Yeah, also a great question. So sure, the XHGLs are fluorogenic, and as we showed, they can be used for paint and minflux, and certainly the fluorogenicity will help to suppress background signal from the unbound fluorophores. What I cannot comment on, because we did not investigate it, if the spirocyclization of the rhodamines, and this has been also used to do single molecule localization microscopy studies, also contributes to the blinking. So we use the blinking just by the binding and unbinding, but of course there's also this fluorogenic effect that helps and can be further explored. Yeah, speaking about HaloTag for paint, so overall I think that the beauty of self-labeling proteins is that we can use it for live cells, right? And this is something that what so far was quite difficult for paint and minflux because the acquisition times are very long. But there has been some recent studies that used deep learning algorithms in combination with the exchangeable HaloTag ligands to also do paint with a high temporal resolution in live cells. And this is where I see a bright future.
Okay, thank you. So maybe we have time for one last question. And there was a question if you can comment on the nuclear permeability of the HaloX probes.
Mm-hmm. So I've shown a lot images of H2B, histone 2B staining, so live cells that has HaloTag in the nucleus. So there's definitely a good permeability to the nucleus. We did not quantify this, but to me, I don't see any problem at a working concentration of 100 to 500 nanomolar to stain something that is localized to the nucleus.
Okay, thank you for answering all the questions. So I think that is what we have time for during our live Q&A. So if your question has not been answered yet, we will contact you by email later. All your questions will therefore be answered, and the chat box will remain open for another 15 minutes. So if you have any follow-up questions that you want to ask, feel free to add them to the chat box. We will answer them via email or contact us via email. I would like to thank Julian and Florian once again for answering all the questions and the nice talks. And I thank all of you for staying with us. Have a nice day.